Welcome to the tutorial for PyProfiler, which is the software for analysis of the data obtained by single particle profiler technology. When you download the software, it will appear as single file distribution, something like this. And in order to open it, you just need to double click. Then you wait a little bit. Usually when you launch it, especially first time, it will be a little bit slow. When the program is launched, it will appear like this. Here you have the area where you will see the list of files you need to process, and here you have some visualization tools, so you will be able to see intensity traces and correlation. First of all, you would need to load the data. In order to load the data, you click the button Import. Here you have to navigate to the place where your files are. And here I will load all of them together. You can select all the files you want to load. Here you can also select data type that you are going to work with. Here I am going to work with the .fcs files, which is the standard output for the size microscopy setup. You click open and you will wait a little bit. The more files, the more to wait. When the files appear in the list, in parallel, you can see for every file over here the fluctuation curves and the correlation curves. For every file, you also can see number of repetitions that were over there. Or here, for example, you have 40 repetitions. And of course, you can look at the traces and correlation curves for every repetition. In addition, here you have number of channels that are present in this data. For example, over here you have two fluctuation channels. In addition, you have two cross-correlation channels. And in order to see less data, you can unhook everything and leave, for example, only one channel, and then only this channel will be displayed. Then what we can do, we can analyze these intensity traces, define peaks, and define intensities for every peak. In order to do that, we're going to go for peak analysis. And here again, the trace is displayed. And if you click here, display peaks, you will see which peaks are detected and which are not. Here also you can see the intensity histogram. You can see the dot plot. And in addition to the dot plot, you can see the general polarization histogram, which actually shows the ratio between values of each peak in every channel. First of all, we are going to analyze the data that was made for these dot plots. And for that, I prepared two types of files that contain data from liposomes that were loaded either with two types of dyes, like over here, LUVs, liposomes with a barrier star red and top floor cholesterol. These are two types of dye in each liposome. And this data was from mixture of two types of liposomes that are loaded only with the single dye. Let's see which types of dot plot would be yield from these two types of data. First of all, we need to define parameters for which we are applying to find our peaks. Over here, there are several parameters. The most important one would be how do we detect peaks and the thresholding. The recommended way to do the thresholding would be the so-called z-score, and z-score is actually displaying how far away in terms of sigma is your peak from the mean value. So it is recommended to work with z-score at least 3. I would go also for 2. It gives usually good data. And here, what you need to do, you can, for example, change the threshold over here. You click Update Threshold, and you see that less peaks are detected now. Then when you go back to 2, now more peaks are detected. In addition, there is an algorithm that will define how do you detect peaks. You can detect peaks based on either one of the channel or on both channels. If you detect peaks based on only one channel, it is pretty straightforward. It is just takes the peak from, let's say, channel 1, and then for every value from channel 1, it takes the value with the same time code from the channel 2. And this will tell you what is the value in the channel 2. Same will happen if you define channel 2 as the channel for detection. It will be just other way around. And of course, you can use both channels. For both channels, there are two types of detection. So-called both or detection will detect particles from one channel and then get the values from the second channel, then other way around, as it is for the single channel, and just combine both data together. If you select both and, you see that the number of detected points drops. And this is because in case of both and, it only takes the peaks that are above threshold in both channels. Meaning that, for example, we detect from channel 1, and then if in channel 2, for the same time point, the peak is also above the threshold, we take this peak. Otherwise, we discard it. So here, if you need to construct a proper dot plot, we need to go to the both or, because sometimes our particles will be not loaded with the dye in the second channel, which means that the intensity will be close to 0 over there. And here, in order to calculate and find peaks for everything, you need to click Fit All. Here you will be able to see some parameters that I will describe a little bit later. What do they mean while the script is running? Okay, now it is done. So we can close this window and go to the window with dot plot. Window with the dot plot is over here. Here, we are interested in these two. These 3 files are for the other type of analysis. We will check on them later. Here we need to select intensity from which channel do we prefer on x-axis. Let's say it's channel 1. Then on y-axis, we will have channel 2. And here we need to select which scale do we want. For example, we want to start with linear. Then we click plot. And dot plot will appear on these two frames. One of them will be just the dot plot. The second will be the density plot. So here you can see that the LUVs that are loaded with both types of dye occupy rather the middle over here, as in, for example, colocalization plot. And LUVs that are loaded only, the mixture of LUVs that are loaded only with one type of dye are stretching along the x-axis, y-axis, or x-axis. To see it a bit clearer, we can go to the log scale. Now it will plot everything again from scratch, but in the log scale. And here you can see this a little bit better. This is the density plot. Right now, this density plot doesn't make much sense because it represents the mixture from both types of data. In order to use this density plot properly, I would, for example, go to only one type of data from one file. Or if you have several files that represent the same type of experiment, you can also combine them all together and it will yield a nice dot plot, a nice density plot. And right now, this density plot actually represents in the log scale what is going on in this type of data. And then if you go to the other one, it will be like this. Yeah. So pretty much these two populations of this density plot correspond to two types of liposomes that are loaded either with one dye or with another. This is how this approach can be used for single particle profiling. And pretty much if instead of fluorescent markers, you use some protein loading or you get your extracellular vesicles, for example, and you want to study how much protein of certain type is over there, you can use this as in kinda nanoflow cytometry. The second type of data where this approach can be used is the data for biophysical profiling. And for this, we have our lipoproteins, HDL, LDL, and VLDL. And of course, everything starts from the peak analysis. And in peak analysis, again, you have your high-density lipoprotein, low-density lipoprotein, very low-density lipoprotein. And here you have again your peaks. But now we are interested not rather in this graph, but in this one, which shows the ratio between peaks in two different channels. For this type of analysis, it would be recommended to use both end, because to have meaningful ratio, we would need to have the meaningful signal in both channels. And again, there is a threshold, and we would need to fit everything. And by fitting everything, we are pretty much fitting this histogram for every repetition, every file. And in this histogram, we are getting, first of all, the mean of the GP distribution, and then sigma of the GP distribution. And of course, the magnitude of the GP distribution. So all parameters will be displayed over here: amplitude, mean, and sigma. And here the fitting will be shown. Now peak analysis is done. So we can close this. And here, if we just click and select these 3 files, and then click plot. No, not this plot, this plot. We will be able to see over here. So there is one outlier over here. And if you look a little bit closer, you'll be able to see that in 3 types of lipoproteins, we have different membrane order. The second parameter that we can extract from this analysis is, of course, the diffusion in solution. To extract the diffusion values in solution, we need to go to diffusion analysis. Diffusion analysis over here. And here we need to analyze these autocorrelation curves. Since for this type of experiment, correlation curves in all channels are pretty much the same, we can analyze just one of them. And here we again have the number of parameters that we can use. And we need to select the proper model, first of all. So we have triplet, we have one component, and we have 3D, because particles are diffusing in full volume. And here, what we can do, we can, again, fix several parameters. For example, we know that offset is 1 over here. We know that our amplitude is 1 because we have only one population. We know that we have free diffusion, that's why alpha is equal to 1. The triplet relaxation is 0.005. And then aspect ratio we need to get from our calibration, as well as we can get from our calibration, for example, diffusion time. And if you want to fit over here, that's what you do. And then you need to do it pretty much for all of the files. So again, you have to click fit all. When fit all is complete, we can again plot over here. And here, in addition to GP, we will also have the plot with the diffusion time. And we will be able to see that VLDL has the highest diffusion time, which means it's diffusing slowest, which is logical because VLDL is the biggest particle and HDL is smallest, so it diffuses fastest. In addition to this analysis, we can again do the dot plot, but for the dot plot, we can use, instead of channel intensity values, we can use these biophysical parameters that we just extracted. So we have diffusion channel 1, and we have our GP. We want to plot it probably in linear scale. And here you can clearly see that they also separate in nice clusters. So green is for VLDL, blue is for HDL, and orange is for LDL. Last but not least, it is important to also show where did we get the calibration values for the diffusion data. For that, I will also import this calibration file. And this calibration file consists only of one channel, and this channel is from the well-known dye Alexa 488, and we know very well the diffusion coefficient of the Alexa 488, but we need to measure the diffusion time to know how our system processes the intensity fluctuations. So for that, again, I go to diffusion analysis. And what am I doing here? Again, I'm fixing this, fixing this, fixing this. I leave aspect ratio. I'm fixing this. I leave aspect ratio and I leave diffusion time. And then I fit this. And it shows 0.028 as the diffusion time and shows 3.7 as the aspect ratio. And these are the values that we are going to use. So now I can update over here the diffusion time that is going to be used. Then I go back to fitting, and now I can correct fitting parameters as I need. So here I measured 3.6. I can fix it. Then I fix this, fix this, fix this, and fix this. Of course, I remove all of the other channels. And we fit over here. Then I fit all. So now I have more precise values calibrated according to the calibration file that I recorded the same day as I did these measurements. So now when I'm going back and I will plot it, the plot will slightly change. And now again, I can go to the dot plot and select Fusion channel 1, general polarization, scale, plot it. Oh, yeah. I didn't select which files do I want to plot, so it didn't plot anything. Yeah, here it is. So this is, again, another possibility what you can extract from these files. Last but not least, you would probably like to save your data somehow, that you can then transfer it to any type of plotting software that you are using. So what you're going to do, here is the output button. You can select the files that you want to output into your directory. Then you click output, and these files will appear in the newly created folder, exactly in the same spot where your source files are. So you're going in there, they will be named the same way as your source files. And if you open it, this will pretty much be the table. You can copy it to Excel, for example. And first of all, you will have your data for every channel. Since we analyzed only channel 1, you will have everything for channel 1, but you will have nothing for channel 2, and then both cross-correlation channels. But then you will have everything for your general polarization data. Another possibility to output something would be, for example, over here in your diffusion analysis, where what you can have, for example, you want to show this fit, and save plot data means that you want to save pretty much everything what is appearing over here. And if you click this, then you again go to this folder, you will have the new file that will contain, first of all, the data from your channel 1, and then you will have channel 1 fit, which is your fitting. And you can use it again in your program for plotting of choice. further for your data presentation. The similar thing is also here in the peak analysis. And again, if you choose, for example, a file and you want to plot something, you have over here everything that you can export, and you can unhook. For example, you don't want to see traces, you don't want to export your peaks, you don't want your intensity histogram dot plot, you just want this histogram and plot. And that's what you click. And then again, you go over here. You go to VLDL plots GP. That's where you have your GP histogram and feed of your GP histogram. If you want to analyze your data a little bit different, if you are, for example, not happy with how your repetitions are arranged, and you want, for example, create one continuous trace, you can either use binning over here, which will just add all of your repetitions to one another. And this will create one long trace, as well as one huge GP distribution. This is especially useful if you want to see the full picture of your experiment. And for example, you can output this histogram instead of the histogram for each individual repetition. Also, if you are not happy with this duration of your single experiment, single repetition, if you want to make it shorter, for example, in this file, you have 60 seconds. Maybe instead of 60 seconds, you want 15 seconds. There is a button that allows you to restructure your data over here. And here, what you can do, you can go to the data that you want to restructure, and it shows you how much repetitions does it have. It has 5 repetitions. What you can do, you can put, for example, instead of 25 repetitions. And you apply it, and what you can see, that now every repetition is, instead of 60 seconds, 12 seconds, because you made them 5 times slower. You also have some sort of correlation function, but because this correlation function is only based on this data, it goes only 10 to minus 3. So it is not the best data for fitting of the correlation functions and extracting the diffusion parameters. However, you can do your peak analysis differently over here.